Welcome back to this explainer, everyone. Okay, so we've all heard the basics of genetics, right? Usually starting with Gregor Mendel and his really famous pea plants. Mendel gave us the foundational rules of inheritance. The idea that dominant traits mask recessive ones and that genes sort independently. But here's the little secret that makes biology so incredible. The real world is far messier and honestly, infinitely more fascinating than Mendel's simple peas. Today, we're looking at the rule breakers. We are going to explore the deviations from Mendelian inheritance that explain the brilliant, complex reality of life around us. Let's get into it. Now, before we look at how nature actually breaks these roles, let's just do a super quick refresher on our main player here, the allele. Think of a gene as a broad category, like, say, flower color, and an allele as the specific version of that gene, like purple or white. Crucially, these alleles occupy the exact same location, or locus, on a homologous chromosome pair. Basically, every individual has two alleles for each gene. Keep that in mind, because it's exactly this relationship between those two alleles that is about to get very, very complicated. So here's our roadmap for today's explainer. We're going to hit 5 main rule breakers. 1. Incomplete dominance. 2. Codominance and multiple alleles. 3. Polygenic inheritance. 4. Sex-linked genes. And finally, 5. Linked genes. All right, let's kick things off with number 1, incomplete dominance, also known as the blending mystery. Now, Mendel would absolutely tell you that if you cross a dominant red flower with a recessive white flower, well, the red completely overtakes the white, right? You just get all red offspring. But let's look at what actually happens when we take these two specific parents, a homozygous red snapdragon carrying two CR alleles and a homozygous white snapdragon carrying two CW alleles. What happens when these specific alleles interact? Boom! Instead of a red flower just completely masking a white one, they actually compromise. We get an entirely new intermediate phenotype, a pink snapdragon. It's kind of like mixing paint. Neither the red allele nor the white allele is fully dominant over the other. And it gets cooler, because when these pink F1 hybrids are crossed with each other, their offspring yield a 1:2:1 ratio. That's 1 red, 2 pinks, and 1 white. It is just a beautiful demonstration of incomplete dominance in action. Moving right along to number 2, codominance and multiple alleles, the blood type mystery. So if you've ever donated blood or even just know your own blood type, you've literally experienced this one firsthand. In standard genetics, a gene usually just has 2 alleles, but for human ABO blood groups, a single trait is determined by 3 different options floating around in the population, IA, IB, and a lowercase I. Both IA and IB are dominant, expressing antigen A and antigen B, respectively, while that little I allele is recessive and expresses 0 antigen. But here is the really fascinating twist. IA and IB are codominant. That means if they happen to end up together, neither one backs down. They are both fully, equally expressed. So, how does this actually play out for us humans? Well, because we have 3 alleles available, there are 6 possible combinations, or genotypes, that you can inherit. But those 6 genotypes only manifest as 4 distinct blood type phenotypes: type A, type B, type AB, and type O. Take the AB phenotype, for example. Because of that codominance we just talked about, someone with this blood type proudly displays both antigen A and antigen B right there on the surface of their red blood cells. It's just a fantastic example of multiple alleles and codominance working together. Now, really quickly, before we go any further, it is incredibly easy to get multiple alleles confused with our next concept, which is polygenes. So let's clarify that right now. Multiple alleles, like our blood types, means there's just one single gene locus, but there are 3 or more versions of that specific gene available in the whole population. Polygenes, on the other hand, means one single characteristic is controlled by the additive effect of 2 or more entirely separate genes. Which perfectly brings us to number 3, polygenic inheritance, or the spectrum mystery. Okay, we are definitely stepping up the complexity escalator here. We're looking at traits that don't just fit into neat, clear-cut categories, like blood type A or B, but rather they exist on a continuous spectrum. Take human skin color. It is not controlled by a single gene. To simplify it a bit, we model it using 3 separately inherited genes. Let's call their dominant alleles capital A, B, and C, which contribute to darker skin, and recessive alleles lowercase a, B, and C, which contribute to lighter skin. A person with the genotype capital A, capital B, capital C would have very dark skin, while a person with lowercase a, lowercase b, lowercase c would be very fair. Most of the population actually falls somewhere in the middle, creating this incredibly lovely bell curve of continuous variation. And to make it even more fascinating, environmental factors, like how much exposure you get to the sun, also interact with these genetics to affect the final visible trait.
All right, number 4, sex-linked genes, the skipping generations mystery.
So, human sex determination relies on the X and Y chromosomes. We know females have 2 X chromosomes and males have 1 X and 1 Y. But just look at the physical difference there. The Y chromosome is much smaller than the X chromosome, and it carries far fewer genes. The X chromosome, though, holds tons of genes that have absolutely nothing to do with biological sex. Because males only have 1 X chromosome, they are hemizygous, basically meaning they only carry one single copy of those X-linked traits. Let's put this into perspective by looking at hemophilia. It's a sex-linked recessive disorder where blood clots really slowly. A female needs to inherit 2 defective recessive alleles, so 1 on each of her X chromosomes, to actually be a hemophiliac. If she only has 1, she's just a healthy carrier. But because males only have 1 X chromosome, a single defective recessive allele guarantees they'll suffer the effects of the disease. There's literally no second X chromosome to act as a backup and mask it. This is exactly why X-linked recessive diseases overwhelmingly affect men. And actually, it's the exact same reason why red-green colorblindness is so much more common in men than in women.
Finally, we reach number 5, linked genes, the packaged deal mystery. Mendel's law of independent assortment basically states that alleles for different traits sort completely independently of one another during gamete formation. But honestly, Mendel was pretty lucky with his P's. When genes for 2 different traits are located physically close together on the exact same chromosome, they're called linked genes, and they completely defy independent assortment, simply because they are physically attached. They are inherited together as a single packaged deal. But, biology always has a trick up its sleeve. And nature's ultimate remix here is crossing over. During prophase I of meiosis, these homologous chromosomes literally cross over and swap physical pieces with each other. This breaks those links and creates brand new recombinant gametes. It is an absolutely vital process because it dramatically increases genetic variation within a population, effectively ensuring that no 2 offspring are exactly alike. And that brings us to the end of our explainer today. From the beautiful blending of incomplete dominance in snapdragons to the co-dominance of our blood types, that continuous spectrum of our skin color, the really unique vulnerabilities of sex-linked traits, and of course the physical tangles of linked genes, we have seen that the true brilliance of genetics lies in its messiness and its complexity. Mendel gave us the alphabet, sure, but these deviations, they are the poetry of life. So I want to leave you with this thought today. If these rules barely scratch the surface, what other incredible genetic mysteries are just waiting to be decoded in our chromosomes? Thank you so much for joining me. Keep asking those questions, and I'll see you next time.